Okay. Hello, everyone. Thank you for coming. It's a pleasure to be here. My name is Mauro Rebelo. I'm a marine molecular biologist. I'm representing here a startup company, which is a biodiversity tech. So we have been working for the last 15 years now, developing technology for biodiversity measurement and conservation in Brazil. And well, we are very excited with this movement of biodiversity credits, and we have been in Brazil trying to show to the even to the supply side or to the conservation projects in Brazil, and also to the demand side, to everyone interested in finance these conservation projects, how we can use environmental DNA to measure biodiversity at scale, right? And maybe filling in a lot of gaps that we have in biodiversity in general. So today we are not going to focus so much in the eDNA measurements themselves, but maybe in a step forward, in a step later, which is how to try to verify this data in a way that will bring more credibility and reliability to build the biodiversity credits. So in a way, our the thesis that we are trying to validate, and I invited my friends Guilherme Oliveira from Vale Technology Institute and David Canassa from Reservas Votorantim to discuss this in a very informal and open way here with you. So please even feel free to ask questions to any of us to discuss what are these challenges in related to the biodiversity measurements and the reporting of these measurements in a way that will fill in these gaps that we still believe that we believe they exist to kick off this biodiversity credits market. Our belief is that there's still a lot of uncertainty in the measurements, especially because most of the projects still work with traditional measurements of biodiversity that are capture based or that are morphology identification based, and because of that they face all the limitations of scale up, right? It's very difficult to scale up these measurements, and we believe that they don't tell the whole story. So they create the current paradigm in biodiversity identification is still the morphology one, and eDNA still hasn't been fully accepted. But you know, biodiversity is vast, and we have to. It is a big data problem. It's not a small data problem. So using a small data approach to a big data problem, we know it doesn't work, right? And one of the advantages of using DNA is that it's a digital asset, right? So it's very easy to digitize DNA. You go from the chemical structure, from the biological sequence to a data file that can be further processed in a very informatic way, and even protected with cryptography. So we want to bring all these points. There are some buzzwords here, right? So blockchain and big data. But the truth is, we haven't been using the latest technology to measure and to report biodiversity. And there is the potential to use much better technology than the current paradigms to address this big problem and bring more credibility to the measurements. So maybe I would like to start asking both Guilherme and David, instead of the typical, can you introduce yourself briefly to talk like, do you see the projects that you are involved as a supply or as a demand in terms of biodiversity, supplying biodiversity measurements or credits, and as a way to introduce you and your theory. Thank you for being here.
Thank you so much, Mauro. It's a pleasure to be here with you. And this seems to be a very hot subject. The room before was impossible to get into, and it seems like there's a lot of people interested. So that's good news. So we are kind of on both sides because the Vale Institute of Technology is a private non-profit research institute maintained by a large corporation. Vale. And so we work closely with the company, and we are using eDNA to access biodiversity as a monitoring tool at industrial sites. We are using eDNA as a way of evaluating land that's been reclaimed, both post-mining and also places that are being revegetated due to degradation for other reasons, such as pasture land, and also in association with the carbon market. So you can say more about carbon than just carbon. So biodiversity information could either add a bonus to that carbon that's captured, can be monetized, or even if not, people can know that you're doing more than just planting whatever trees. And we are not in the market for doing this, but we are using this as a tool to assess biodiversity and creating measurements such as measuring impact of companies and how companies can act to minimize that impact and even sometimes gain and have a no net loss or net gain aspects of it. So we're kind of both sides, but not as a business, more as a research institute. So I agree with everything you said. And what we do that is not usually noticed is that to add value to the eDNA, you need good reference libraries. And unfortunately, these are not readily available for Brazil. So we are very much in the business of creating those libraries and putting this technology to use, both by, oh, I wasn't here, both by comparing eDNA data with what has been measured using the gold standards, which is people on the field collecting information. And I can get back to what these results show. Yeah, this thing screwed me off. And and creating the reference libraries for every place we go. So currently we have the only complete flora described for any region in the Amazon, and associated with that flora, we have the DNA barcodes for every plant. So we are in a very good position to test this technology in a field that has been really well studied. So I'll stop here for now and pass it on to my colleague for some comments. Thank you. Microphone dance.
Hello, everybody. I'm David Canassa. I'm the CEO of Reservas Votorantim. And Reservas Votorantim is a company focused on managing forests in several biomes in Brazil. About your question, first, of course, because we manage territories, we understand that we can supply many of the possibility of credits that could be created in the next years about the biodiversity. When you're thinking about Brazil, we have a lot of job to do yet. For instance, together Biobiro, Legado das Águas developed the first project to mapping part of the biome with the DNA before the idea of the DNA. And when we did this several years ago, 7 years, I think nobody thinking to map the DNA of the Atlantic forest yet. It's incredible. If you're thinking that Atlantic forest is similar Amazon regarding the quantity of biodiversity that we have, then we can imagine it, that we have many solutions inside the forest that we don't discover yet. When you talk about another biomes like Cerrado, for instance, that we are developing a project since 2015. In your first mapping in this biome, we discovered 7 new species. with 1 month of studies. Imagine that we have in Brazil yet. Then the idea of the to mapping all DNA of the our biomas, not about the plants, but all all species that we have in the forest. It's so important to not only to create a possibility to credit, but if you are talking regarding the probably the biodiversity is a problem much bigger than the carbon. But the people don't understand this aspect yet very well. And of course, we have a connection between carbon and biodiversity. It's for us in Brazil, it's clear that it's connected. Perhaps in Europe, perhaps in the United States, not so clear. But for us, it's clear. Then we need to spread this idea of implement the eDNA could help us to not only to maintain the forest, valorize that we have, and probably discover many species that imagine if you take one cup of water in any reservoir in Brazil, when we pass for one scanning in the DNA, how many species probably uh, we didn't have any register yet. This creates the possibility to discover many things and perhaps some solutions for our problems regarding medicine, regarding biofuel, and so on. And I believe that this could be a it's very important we spread this idea and implement this idea not only in pilot initiatives, but around all Brazil and South America, because we have many areas that we need to understand better the biodiversity of these areas.
Thank you. So when we working with eDNA, we try to talk to a lot to people on the demand side of the biodiversity, right? And we have been facing the following challenge. One, um, not everyone really understands it because it's so complex. I think that the people have been painting a picture of complexity to biodiversity that it's real, but creates almost a challenge that it's impossible to solve, and that cannot be true, right? So there is a challenge of simplifying biodiversity to the point that it's a manageable problem, right? But not far from the complexity that we find in the field, but it has to be manageable. Otherwise, people simply abandon it. And on the other side, there is, because it's so hard to understand that there's so much uncertainty connected to the measurements, there's a problem of trust, right? So, do we trust this? Can I trust this? Can I put my reputation attached to these measurements? So, many times we've been trying to see, or we are perceiving this trust problem as the main problem. Not sometimes it comes in the format of, for example, when you say, "Wow, we can use DNA to measure," and then people say, "Okay, but who is going to certify?" So many times the certification question comes, and it's a way to demonstrate that, well, I don't understand this, I don't really know, like, I don't even believe you, so I need someone else to certify this. And we are so we tried to address this particular problem. How can we bring trust and confidence to the measurement, to the point that the client does it's not worried about who is going to certify, right? And unfortunately, the technology that can provide this which is blockchain, is in itself complex and many times hard to understand for a lot of people. But this is what we have developed, and you'll be able to take a deeper look at our measurement, report, and verification solution outside of this conversation, because what we tried to address is exactly this problem of trust. So DNA, eDNA, and my opinion, and we have, it's inevitable because of the large scale of the measurements and of the conservation that we have to do. We won't be able to tackle this problem without a technology that is scalable. And DNA sequencing, the technology of DNA sequencing, is one of the fastest growing technologies in human history, right? So has evolved 10 times faster than Moore's law for the silicon chip. To sequence a human genome in 2001 costed $100 million, and now costs less than $1,000. So it is, if you have a biodiversity problem and you have the chance to address this with the technology that has, like dropped in price more than what, 100,000 times. So that's the right technology. So it's very easy thinking from first principles, trying to leave ideology and maybe even politics out, that DNA sequencing is the right technology to address a complex problem like biodiversity. And if we choose this route, then it's possible to create a kind of digital twin of biodiversity. So because you can digitize everything, then you create a digital twin of the environment. And if you are able to do that, then we have something that is a digitally native asset, right? And if you can do that, wow, then you can apply cryptography, bioinformatics. You can do a lot of stuff with this digitally native asset, and that's what we have, what we've done now, right? So by taking the environmental samples that are full of DNA from the biodiversity, sequencing it, you create this digital twin, and then applying cryptography, we get to the tokenization of this asset. But it's more than just creating a token of a credit. We are actually tokenizing the digital twin, which means now you have uh, um, information that can back any claim about information that cannot be tempered, right? And you can back any claims, biological or ecological, from an asset, because you can go, like looking at from hash to hash to hash to hash, back to the raw file that comes out of the DNA sequencer that gives you the proof of the integrity of the data. And this is very important because trust is based on integrity, right? And we believe that this trust problem is the one that will help not only uh, DNA to be accepted as the main technology for biodiversity measurements, but also to kick off, to solve the trust problem and kick off a biodiversity market. When you think about the coming Montreal mark, um, they put the necessity of we create the biodiversity credits, but give us some rules to do this. For instance, what is the dependence, for instance, of that place regarding the investments and so on, or the risks. How we today do this? Monitoring in the field, using many people with spend a lot of money, and the truth of this information normally is difficult to verify because, um, of course, um, we can create campaigns for 2 or 3 years to monitorize all the data. But if, instead of this, we bring the possibility to, with one sample, uh, we have the one very good idea of that we have in that place, and after we can send or yes compare with the other results. Imagine how more fair this process could be in the future. Um, then the idea of the EDNA could, in my opinion, help us to decrease the risks of the implementation of many projects before the creation of the credit, for instance, and give us be more confident to implement some actions in that regions. Um, because, of course, in case of Brazil and other countries have forests yet, of course they need to develop your economies. They need to use the biodiversity, but if you understand better the biodiversity, you can decrease the risks and understand better the dependencies of the region. And to do this, we need a better information. And I believe that EDNA could be a source of this information.
Yeah, it is a great source of information. But going back to the trust issue and taking a step back, um, how do you trust that this works? How do you trust that the plants and animals that you find on EDNA are the actual plants and animals that are in the field? So that's why we need to do this work to demonstrate on places that were studied with boots on the ground that we find the same things. And we've done this work. It's about to be published in the next couple of weeks in the forest of Carajás in the Amazonian region. And what we find is that, well, no technology will see everything, okay? So not even people on the ground or not even eDNA. But we do see further, we see a whole lot more species than by having boots on the ground, both animal and plants, and also soil biodiversity. We see it more consistently, meaning that if you have a person, parataxonomist or taxonomist on the ground and collecting samples at certain points, the variation is bigger than what we see with eDNA. Okay, so because we see a whole lot more, but of course in the eDNA world we miss a few things because some species are difficult to sequence, and we keep the primary development is something that's ongoing, and so we are pretty much happy with the results that of the consistency with which we identify species. Second, whoever is doing this work wants a list of species. So also that creates trust, because if at the end of the day you don't have a good reference library, what you end up with is what we call operational taxonomic units. So that means that we have a bunch of sequences that look like each other, and they represent an entity that may be a close relative to this animal or to that plant. But you can't really assign a name to it. It allows you to create interesting metrics on the level of biodiversity at this space compared to the other space a long time. So it's still useful information. But we would like a name. Is this an endangered species? Is this an invasive species? And other types of information. So completing these reference libraries, I think, to me, is part of even from a strategic point of view of the country that wants to be a player in the biodiversity credit market, we need to have names. So the scientific community mainly needs to be empowered to create these libraries. Second, on how you follow up on the data that was produced. I'm a firm believer that even companies should disclose this data, because it is important then to populate the public space with the species diversity at each site that is analyzed. Okay, and there are public repositories for it. So there is GeneBank, there is Eyeball and Brave, and now GBIF is coming into this scenario as well. Of course, technologies like blockchain can add another layer of traceability to this data, But we have in place already some instances that allow one to see what was produced. And if I don't trust his data or he doesn't trust mine, I can go and reanalyze it. And that's the beauty of it, what Mauro said, that as the libraries grow, we can reanalyze data and have better information. And that's just impossible, or it's very time consuming to do it with specimens that are deposited in collections. Okay, that is important because doing that allows you to create reference libraries. And for instance, we're going now to collections and extracting material from specimens that are already deposited there to create the references for those species. Okay, so I think that that adds all this effort adds to the how trustful eDNA is, and I think that I totally agree for the area that we need to study. I see it hard to use a different technology. Just at our site, it's 800,000 hectares of forests. In addition, the company is protecting another 400,000 hectares and reclaiming another 100,000. So I don't see any other way of monitoring such large spaces. So and creating trust will be important for the and finally to is how the environmental agency looks at this. They know that there is an eDNA, they know that it is useful. So we partnered with the Brazilian Environmental Agency in a project to use the eDNA to monitor the national public parks in Brazil. So this is a way for them to understand the technology, get their hands dirty with it, know what it can do and what it cannot do. And then when other companies who want to monitor the sites or provide a service come with the eDNA data, they will understand what this is all about.
I have mixed feelings about the reference libraries, but it comes to mind. Francis Crick once said that it took 25 years for the double helix of the DNA to go from a crazy idea to the obvious truth, right? So imagine it, how something very key in biology. Imagine it's understandable that the DNA it's taking time and it will maybe take more time. Going back to the size of the areas, so we know like if you take all carbon projects in Brazil, they today are around, please correct me if I'm wrong, people like around 10 million hectares. That's super too large. How can you guarantee that this carbon it's actually have an impact in biodiversity with even if you consider camera traps or sound recordings just too large. We need so this thing, I have a firm belief that it's inevitable. Okay? And if it is, and assuming that we don't know most of the species that exist, I think that the connection with the name, even though it's very important for our human brain, it's not necessary from the machine point of view to analyze all the data, and it may end up being a barrier, right? And what I can see from the challenge, I don't want this discussion to become very technical, but today we are giving names passes through the selection of primers of probes that we use to look through all the DNA. And here comes a lot of variability because you cannot in the end, I think that what we need is to have more genomes. You can give a name to the genome or not, but you need the whole genome so you know that every piece of the environmental DNA where it fits to to an organism, right? And this I believe will be key for this to move on, even if we manage to move from name or no name. Still believe names are important for people, but not necessarily for us to see this reality about the environment. So what I think is, so I want to go back to the transparency that also we talked about. So I see transparency in two ways.
Yes, there are a lot of companies that still don't want to disclose their information, which is somehow silly because now you have citizen science, no, science citizens that are doing eDNA by themselves. The cost of analyzing one sample it's around $100 to $200. So like people, I live by this lake. They go there, they get a sample, and they analyze the same way that you can ask 23andMe to analyze your genome. So like people are analyzing now the environmental DNA of their backyards or of, where they live. So the information is out there. And if you, I don't know, have a are the company imagining a hydroelectric power plant and you have a reservoir. So believing that no one will know that it's silly because everyone will know, even if it's just like people that live around. So there is this transparency about disclosing the data, but there is a second level of transparency, which is guaranteeing the integrity of this data in a way that people will know if you try to modify the sequence files, or if over time you try to delete. No, or no, maybe in 10 years this information is not in my interest. So you can try to pretend it never happened. So we now have tools with these digital things with tokenization to create proofs of existence of a biodiversity profile in space and time. And this is what we believe will, over time, also create, will help the companies that are really interested in in preserving the environment to get a reputation that's unassailable. Why? Because all the record of their efforts it's now protected in an immutable record on the blockchain, and all the information is disclosed and can be analyzed by anyone. So the eDNA, there's so much that we can talk about this, right? But I'm wondering if you have uh, any comments on this aspect of the reputation of the companies that want to demonstrate their commitment, not just with the environment in terms of carbon, but with the impact of this carbon or of the carbon project on the biodiversity. How do you see this, the risks attached to the reputation of connecting their brands with measurements?
Well, Reservas Votorantim developed the first project in Cerrado with Terra. And of course, we implement the CCB uh, certification. But when I studied this case to implement the CCB, uh, um, if we are talking about carbon, what species are important to maintain carbon? CCB not will answer this question. It's very interesting. Normally, we um, decide some very good species, for instance, a monkey, butterfly, or someone. And this is the criteria accepted today. Okay, if we decide one criteria that's not connected directly with biomass, for instance, how we can measure this criteria along these years? Okay, we today we contract very people, much people to put trap cameras and so on to solve this problem. EDNI can help us in this, of course, but I believe the solution is different. Um, Here in the biodiversity copy, we are talking about the connection between carbon and biodiversity. Uh, In your reserve called Legado das Águas in São Paulo, we developed a new methodology with carbon that will try to answer this question. What is the aspect in biodiversity that's more contributive to maintain the forest, the biomass? Then you try to measure exactly this aspect. For instance, in Legado das Águas, we have a monkey called Muriqui do Sul. It's the gardener of the forest. If I lost this species in Legado das Águas, I need to contract many drones to spread the seeds in the forest. This one very hard aspect that we need to measure and try to maintain this species in that territory. EDNA could help us in this, of course. Of course, we need to maintain the researches together to see the species, but it's one example how we can connect the things. We need to connect the carbon and biodiversity programs. If you understand very well what we can measure in that region, what's the most important aspect, we can develop methodology to do this. And along the years, we can verify if it's true that forest is increasing and the species is increasing too.
Excellent. Well, that has been demonstrated for elephants, that they help to fix carbon just by spreading seeds. And we're testing that now with tapirs in the forest because they are the actual gardeners of the Amazon forest. But other species that are seed dispersers, such as bats and birds, pollinators, and you can use eDNA for that too, because it's not only a metric of the species that are present. You can use eDNA to get information on their relative interactions. For instance, we can sample flowers to see which insect species visit those flowers. And you can sample bees to see which plants they collect the pollen from. So it creates this network of interactions, and you come to this issue of which species are important for maintaining this network that increases ecosystem viability. And also with bats, which plants bat visit, which insects bats eat, we can sample their one-off and understand that. So it's eDNA has more uses and increases the knowledge and the interactions and the ecological perception of that ecosystem. The other point, going back to your transparency issue, is on methodology, analytical methods. Um, I can't take some any metrics that I don't know how that was analyzed. And we have plenty of methods that are public, robust, and I think you use them, we use them, we create our own pipeline and you do yours, but we need to disclose what this pipeline is. Otherwise, how do I know that this name attached or just an OTU ID attached to the sequence is properly attached. Which library did you use to compare your initial sequences? So there are other steps in bringing transparency to this field that to me is important, but I realize I come from a more academic perspective to this issue. But to me, data release, transparency, and how you analyze this data, the libraries you create is important. And we can go on discussing reference libraries, but we can have a coffee over it.
Yes.
Those are the two points I want to make.
Thank you, Guilherme. So, we have like for my more 5 minutes. I wanted to ask if anyone in the audience has a question. Yes, we need the microphone. So,
Thank you very much. It was a really great presentation. There are a lot of teams and uh, like approaches that are quite familiar to us. I'm the chief scientist of New Atlantis Labs. We are focused on marine biodiversity markets and sustainable finance mechanisms. And we use not only DNA but also metabolic approaches together with ecological modeling in order to build digital twins. So my question is, whether you guys using more sophisticated techniques that are also like molecular ecology based approaches in order to go beyond mere species count. And the other question is, are you using a private or a blockchain technology, blockchain technology? Because this is absolutely critical
Yes.
in order to create liquid markets for nature. Because otherwise, at the end of the day, we want to create demand for this type of assets.
Yes.
Not only carbon markets, but actually biodiversity based markets, which are the real productive assets.
So the first part of the question, we are not using more sophisticated measurements. Actually, I'm betting on the opposite, that richness is going to be the key, the most important measurement, and the one that we can rely to do to answer most of the questions, right? So richness of OTUs. That is my bet. The way that we are trying to give more transfer, of course, we can do, all that we want. There are, like he said, we can measure different levels. But my second bet is to go leave the primers and go shot. This is technical, okay, but shotgun the whole eDNA. So, kind of, the answer is more sequencing. So if you increase sequencing depth, you have so much information that, and this is just about cost, and cost of DNA sequencing keeps dropping. So if I look the next 5, 10 years, I think we are not going to work with primers anymore. We are going to shotguns, exactly what happened with the human genome. So, but this is a bet, right? And but I believe this will allow us to look at simpler metrics to answer most of the questions. With the what we are trying to do is to create a machine-friendly report of all the data, including the scripts and the pipelines. So you create a JSON file with all the URLs to the raw data, with the hash of this data, and with the scripts that, everything is in that JSON file. So machines can talk to each other and analyze and do everything. And your last part, we are using the Liquid network, the blockchain of the Liquid network, which is a side chain of Bitcoin. So it is, if in terms of safety, is protected by Bitcoin, and it's a public blockchain, and we can talk more about this if you want.
So as other tools, yes, we are using many other tools, not necessarily because of biodiversity credits, but including, for instance, we're working at a site where there's a bird species that in nature there are 20 birds known. So you could think of attaching biodiversity credits for protecting that one species in addition to all the biodiversity in that place. So we have genetic variability of that species in that environment. So we're doing genomes for endangered species. We're doing in large scale. So and in places where you have endangered species, that also can be a bonus potentially. The other way we're doing it is looking at soil quality. We're using metaproteomics instead of using DNA. We're using proteins, and that gives us a very clear picture of all of the metabolism in the soil and the microorganisms and plants and nematodes and so on. They're doing that metabolism. And so, I mean, these are two examples of other technologies that we are deploying, not necessarily because of credits, but to assess other types of information in a certain sense.
So our time has run out. Thank you, David. Thank you, Guilherme. Uh, okay. So if anyone has one more question, then No, thank you so much. Then, yes, please.
Hello guys, congratulations. It was wonderful to be here with you. Actually, I'm gonna skip a little bit of the main issue that we'll discuss here. Okay, we are Brazilians. I'm on behalf of Brazilian Association, Intellectual Property Association here in top. And I think that we have two big companies here, and I'd like to know what, in your opinion, guys, what what we have to do, what is the government had to say, have to do, what we have to do to create more situations about like because everybody knows not only Brazilians but also foreigners that Brazil is a very potential. Okay, it's a very not only Amazon but the biome that you work to, and I believe that we have to give next step to create more situations about use DNA, about use blockchain, about use technology to create to become not only a potential, but really, really lead this new issue. What do you think about that? Well, we have two companies here that are doing this. Okay, Reservas Votorantim started this almost 12 years ago, thinking about the possibility of the biodiversity. What I think, what the situation today? In Brazil, we have a very good law, biodiversity law, but normally the companies don't understand the law. They have fear to use the biodiversity. Uh, we learn about the law. We are using, we are developing projects, and we only need to spread this idea. We have many possibilities in Brazil with this. Regarding EDNA, it's very interesting. We have so many institutes in Brazil, uh, uh, universities and so on. We have money to develop research, but nobody think together to create a very big database regarding Brazilian biodiversity. We need to develop this. I don't have the answer. I know what I'm doing along these 10 years. And I understand that we need to talk more in Brazil about the law. In my perception, it's the first uh, problem that we have. The people need to understand what we are talking about. And with this, we can perhaps uh, change the potential in business. I believe in this.
Good answer.
Well, clearly, everybody recognizes the value and the potential of the genetic resource that countries that have untouched biodiversity maintain. So, how do you translate that into business? The thing is that at this point, only developed countries created, let's say, large-scale genome projects to directly assess this genetic resource and then transform it into business. And the human genome is a very, very good study case because it took $3 billion to do it, and the return on investment was higher than anything you can think of.
1 trillion.
Yeah, so we need to sequence our biodiversity, and for that reason, we started a large-scale project in partnership with the environmental agency to do the. It's a large scale, but our diversity is super mega large. So we need to partner with other companies, and also. I think it's a matter of national strategy to do this, and the government should be much more into this than it actually is.
I will just add that the financial resources in Brazil exist. So if you take the mandatory investment in R&D attached to the concessions in oil and gas and in electricity, this is around $2 billion a year. So the money is there. It's just about the corporations to make the decisions and not wait for the government. Because I believe the government said, look, the money is there. Why you're not doing? And the technical capacity
Brazilian has the scientific infrastructure and minds. They are there. It's just deciding. Thank you, everyone, so much for being here.